Good morning, good afternoon, good evening to nearly 200 people across the world and who are also watching the webinar as a recording. I'm Jennifer Smilowitz. I'm the Director of Scientific and Strategic Development of the International Milk Genomics Consortium, or IMGC, and I am excited to bring to you our annual Spring Roll Up One Sleeves workshop. This workshop is hosted live, but it is also recorded. And the recording will be available to you to view on the Milk Genomics Consortium website found on the left-hand side of the screen. We'll post it by July 1st of this year. Before we start the workshop, I'd like to share with you a little bit about the IMGC. Many of you know our origin story, but for those who do not, we are a scientific society created in 2004 by Professor Bruce German at UC Davis and colleagues from all over the world to make one discovery, and that is to sequence the lactation genome, which the group accomplished in 2009. But today, the IMGC is made up of hundreds of scientists and innovators worldwide from academic and industry sectors who work on the common goal of advancing the field of milk science to improve health. So we advance the field by leveraging 4 platforms. The first is our flagship annual conference that's hosted in the fall and features scientific cutting-edge research and accelerates scientific innovation. Our symposium attracts a diversity of disciplines and fields, fosters collaboration, and increases the impact of publications in milk science. The second platform is our awards program, which highlights scientists across career timelines from students to legacy pioneers in the field. And this platform encourages ongoing engagement with our base. The third platform is our bi-monthly publication called Splash Milk Science Update, which translates the latest scientific discoveries in milk science from primary scientific peer-reviewed literature written for a broad audience. We also leverage professional scientific writers to write all of these articles. And we have published more than 450 articles on milk science that are searchable on our website. And finally, our fourth platform applies to today's event, the annual Roll Up One Sleeves, or ROS workshop. This is a practical 3-hour focused webinar that addresses translational issues specific to scaling up, analyzing, and navigating regulatory for milk components. And the goal is that participants come away with a better understanding of bringing discoveries to practice as concepts or products or services for human health. Now, based on data from the exit survey completed by attendees from last year's workshop, this year's topic is milk diagnostics for lactating parents and babies. And this is a timely topic as the FDA has recently launched Operation Stork Speed. This is an initiative to evaluate the safety and nutritional adequacy of infant formulas. Now, understanding the composition and structural complexity of human milk is fundamental to support a human milk diet and health outcomes in the maternal infant dyad. Now, to help us select next year's topic, whether you are part of our live audience or watching this webinar as a recording, please take 5 minutes out of your time to complete this exit survey with the QR code shown on the left-hand side of your screen. The workshop is hosted as a webinar with back-to-back presentations from world experts in chemistry, nutrition, engineering, and medicine. And today's featured experts include distinguished Professor Carlito Labria at UC Davis, distinguished research scientist Dr. Lindsay Allen at the USDA ARS, physician scientist Dr. Nicole Cacho at UC Davis Medical Center, and Professor Timothy Chan at the University of Toronto. Now, in their presentations, they will share with you their experience, their expertise, and insights into the challenges that they've experienced and solutions that they propose to overcome those challenges in milk diagnostics and therapeutics for lactating parents and babies. Now, after the last presentation, we'll take about a 5-minute break, and then the panelists will join in an open panel discussion for about 1 hour, and we'll welcome a 5th panelist, Dr. Daniela Hampel, project scientist at USDA ARS, who co-authored Dr. Lindsay Allen's research that she's presenting today. Please submit your questions or comments anytime during the workshop in the Q&A box in Zoom, which will be included in today's discussion. And without further ado, I am honored to introduce to you our moderator of the webinar, distinguished Professor Bruce German at UC Davis, who is also the founder of IMGC. Okay, Bruce, take it away.
Thank you very much, Jennifer. And I too am delighted to be here. This is one of my favorite days of the year where this organization literally brings together the knowledge, the discoveries from various fields and applies them to milk so we can bring the knowledge to practice. This is my particular favorite subject. It speaks volumes to the priority of babies in the healthcare system that we do not lead the world in the diagnostics of the health of babies. We still rely on discouragingly crude estimates, length, weight, head circumference. We have not brought diagnostics into infant care. And that's one of the reasons to be here today. It is without question the cornerstone of the future of precision medicine to be able to simultaneously measure an individual and treat that individual. The future of medicine is wonderfully inspiring. In order to participate in that, babies will have to have very exact, precise, and annotated diagnostics of their health. That's what we're here for. The first speaker, actually, Carlito Labriglia, a world-famous analytical chemist whose vision is quite remarkable. His vision is to bring chemistry to practice using biology to guide and inspire the directions that chemistry pursues. And his achievements are remarkable. But I want to tell you one anecdote. When we were beginning this project, Carlito Labriglia was a celebrity on campus. He was already a famous chemist. And I went to him and I asked him, we're starting an initiative on babies and especially premature babies. Would you be willing to help? And without pausing, what can I do? And for the last 20 years, Carlito has brought the cutting edge of analytical chemistry into infant studies and milk research. Turns out, Carlito, being a globetrotter, is momentarily out of clear internet connection. So he has recorded his presentation and we will listen to it now. Please take it away, Carlito.
Hello, my name is Carlito Labriglia. I'm a professor at the University of California in Davis. And today I'm going to discuss with you the work that we've done on human milk oligosaccharides. So, as I said, I'm going to talk to you about the glycomic characterization of HMOs. But what I'm really going to talk about is the technologies that made all this possible. And as a reminder, I always like to bring this up. The glycans on blood cells are what distinguishes our ABO blood type. And these are different glycans that look not too dissimilar from human milk. And so there's a blood type, and I'll show you later on that there's also a milk type based on the structures of these different types of glycans. So I'd like to say that the inspiration for this was this Prado Rubens painting in the Prado, which is the birth of the Milky Way, where Hera is feeding Hercules, and she gave so much milk that it created the Milky Ways, created stars in the universe. But really, it was Bruce German in the Milk Bioactives program who asked us a long time ago if we could look at human milk oligosaccharides. So to develop these profiling tools, there were several new technologies that needed to be made. One of those was a new type of mass spectrometer with high resolution and mass accuracy. We needed to develop methods that would separate oligosaccharides chromatographically, methods to annotate the isomers and the structures, and methods to obtain from relative to really absolute quantitation. So for this program, we actually built a whole mass spectrometer. So this was in the late 1990s, early 2000s. We built this mass spectrometer that had high resolution and mass accuracy, mainly to help identify oligosaccharides. So peptides and other metabolites ionized much more easily, and they were persistent background. And so to be able to identify the human milk oligosaccharides from the background compounds, we needed really high mass accuracy and high resolution. And so when I became an assistant professor, I came to build this instrument, and it was really a prototype instrument with an external source for an external source Fourier transform ion cyclotron resonant. It had matrix-assisted laser desorption ionization as its ionization method. So the performance of it, even today, would be considered ultra-high resolution. We can attain up to 1 million resolution with mass accuracy of 5 parts per million. The other unique feature about this instrument was that it not only had collision-induced dissociation like many mass spectrometers, but it had infrared multiphoton. It's ionization, not ionization, this is infrared multiphoton dissociation. And the schematic of the instrument looked like this. Initially we had the MALDI source, but we changed that to an electrospray source. But you could see the resolution of this instrument. This is an oligosaccharide with a resolution of 210,000, which again, by today's standard, would be pretty astounding. But you can see here the C13 envelope, and this is where IRMPD was really useful. This is infrared multiphoton dissociation. Using a CO2 laser, you can take a large compound and we can dissociate it and you can fragment it. And people still use this today to break down large protein, but back then we were already breaking down oligosaccharides with it. So we built this instrument, and this is what it looks like. You can see here, it looks like a home-built instrument. This is a 3 Tesla superconducting magnet. This is the ionization source is out here. This is about the size of 3 refrigerators standing end to end. And you can see here the laser. This is eventually manufacturers or companies started making an instrument like this. And so the next one we actually bought, and this is this one here. The person that I did my postdoc with, where I built a similar instrument, started a company, and this company then started manufacturing these instruments. But even after it was manufactured, we still modified it quite a lot. And so in this configuration here, we opened up the back end so that we could stick a CO2 laser, and that's when we started doing infrared multiphoton dissociation with it. You can see here it's still just laying on a box. This is the controller. And so the second thing that we did after building a high performance mass spectrometer was that we had to develop a chromatographic method for separating oligosaccharides. And the reason for that is that HMOs contain hydrophobic and highly ionic species that are too polar for C18 columns, but not polar enough for helic. And so we needed something that would separate specifically oligosaccharides. And around that time, people were already developing something called porous graphitized carbon. And so we optimized that to look at human milk oligosaccharides, and it turned out it was really the best method for separating them. And Agilent Technologies was at that time creating what they called a lab on a chip. And so we thought that would be a great way of separating oligosaccharides at the nano level. And so we collaborated with them to actually immobilize this PGC in this nano LC format. And so the workflow started to look like this. We'll take our sample, we'll do our separation, send it through this LC with this nano microchip device in it, and then send it into a mass spectrometer. Where we could then profile, and we can get really high limits of detection down to, in this case, femtogram of oligosaccharides. So this was a pretty good method. This is what the actual eventual, this is what the optimized chromatogram would look like. You can see that we can see, oftentimes we'll monitor as many as 300 structures, but it was capable of monitoring 1,000. We would have accurate mass. We could do tandem mass spec, and we could have 3 different methods to characterize the oligosaccharides. We would have the retention time, we would have the mass spec, the very distinct accurate mass, and then the tandem mass spec. So with this, we can also measure abundances. We could measure the fraction, for example, of silated components in those that are fucosylated. So the next step then is, because now we can monitor the structure, we needed to annotate the structure and be able to rapidly identify structure. And so at that time, there were only a few structures that were known, and it would often require a whole PhD thesis to determine a new structure. So what we needed to do was develop a systematic method for structural annotation, in this case using mass spec, MSMS, and we would use enzymes to do that. Enzymes have very high specificity, and so if we had an unknown compound, we would throw an enzyme with the known specificity and use that then to sort of decompose it and determine its structure. We also needed to create software so that we can identify the composition and structures. We were generating millions of data information, and so we needed to be able to sort all this out. And so we developed several software so that we can start looking at these and identifying the different compounds. One of those software that we developed has actually just recently been published. It was called GlycoNote, and what it would do is it would identify compounds for you and determine which compounds or which signals belong to oligosaccharide. But this is the method that we came up with to determine the structures of oligosaccharides. If we had an unknown compound, we would get the mass spec with mass accuracy. We would get the tandem mass spec. We would then throw enzymes at it so that we can get what the linkages were. And then we do a mass spec analysis to see if we produce any products. And then we ask ourselves, do we know the structure after that pass? And if we don't, then we just keep on repeating it using different enzymes until we finally determine the structure. So this is what a tandem mass spec would look like. That would give us some idea what the structure could be. We throw an enzyme in there, and it turns out we wanted to know what this was. We throw in a beta-1,3 galactosidase, and this fell off. We could see that from the mass spec, and then that would give us a structure. So if you do this enough over and over again, what you come up with is many, many structures. And so this was a work by a very persistent and very precise graduate student who's Shuai Wu, who now works for Amgen. And so this is just the neutral oligosaccharides. He came up with another table for the anionic or the silated oligosaccharides. But you can see using this, we can then annotate these structures quite effectively. And so then our workflow would look like this. We could monitor, as I said, hundreds and even up to 1,000 structures, but that wasn't necessary because we found that for each mother, there was only about 100 structures. But if you combine about 5 mothers, we may see about 300 structures. But in any case, you could get your chromatogram like this. The workflow, as I've shown you earlier, was this followed this kind of a format. We've changed our mass analyzer instead of the FDICR, which was not fast enough. We actually used a QTOF in this case, and so it's a lot faster than the FDICR. It's also a lot more robust, and so we can then analyze these milk oligosaccharides. The other thing about this is that we not only could look at milk oligosaccharides in human milk, but we could look at it in a host of other tissues, including including we could see it in blood or plasma, we see it in urine, we see it in feces. So I won't go over those here, but those are all published in various publications. And so when you have a method for looking at human milk oligosaccharides, you can then sort of monitor what happens to them. And so in this case, we were looking at feces to see what happens to the human milk oligosaccharides. And so these are all their abundances normalized to the starting point. And we monitored an infant in this case at various different weeks. So week 1, this is what their milk oligosaccharides look like. At week 2, it starts to rise. At week 4, again, this is in feces, it starts to drop. And in week 12, you can see that it drops even further. And then we monitor certain bacteria, or actually, this is work in David Mill's group. They monitored then the bacteria that was also coming out of the feces. And you can see the blue here is actually B. infantis, Bifidolongum subspecies infantis. And so to us, this was an indication that this bacteria is actually a consumer of this human milk oligosaccharides. And in fact, David's group has looked at this bacteria extensively. This has become even a product. So this is, it has salivase, it has vicosidase. It was extremely well tuned to actually digest human milk oligosaccharides. And you can look at the various different strains of this bacteria. This is a consumption pattern, again, because we could monitor the HMOs. We can give the different strains mixtures of HMOs, and we can see which ones disappear. And based on that, you can see that they have very similar consumption patterns, but there is also variations between the different strains of these bugs. And so this by itself has become quite a big story. B. infantis is known to be a master consumer of HMOs. When Dave's group threw it at different bacteria, at human milk oligosaccharides, it was the highest consumer of human milk oligosaccharides. And other bacteria, for example, E. coli, Enterococcus, they sort of nibbled at it, but this was really the ones but Bifida B. infantis was really the one that was supremely able to break down human milk oligosaccharides. And the rest of the story, of course, as I said, is well known to you. We know that B. infantis was decreasing in the population amongst various parts of the world. We started a company called Evolve that has been renamed Infinite Health, but you can get this bacteria now as a consumer product. So we looked at then the differences between different mothers. And when you're doing human milk oligosaccharides for the first time and you're analyzing it, and you're trying to compare it to different clinical outcomes, one of the things that always sort of gets in your way, or at least you have to be careful of, is that mother's milk comes in 2 forms, essentially, at least in 2 phenotypes, 4 genotypes, but 2 phenotypes. And that is that the secretor and the non-secretor phenotype. So there's a PCA analysis of many, in this case, over 100 milk samples. And you can see that they separate very easily based on the human milk. And so if you look at the human milk profile of the secretors and the non-secretors, you can see that the secretors produce this, and the non-secretors also produce it, but in very small amounts. And other structures are preferred by the non-secretors, whereas there are structures that are produced by the secretors. And so we came up with a method that you can actually just use human milk phenotypes to determine the secretor and non-secretor phenotype of the mother. So this is actually the only method, and even now, that allows you to determine this phenotype based on the milk itself, or based on the human milk oligosaccharide itself. And so we use that, and it's known that secretors, the secretor populations are resistant to some kind of bacteria, while the non-secretors are resistant to other types of bacteria or virus. And so it's important to know whether a milk is a secretor or a non-secretor. And so because we could analyze the phenotype, we received human milk samples from all over the world from our collaboration with the Bill and Melinda Gates Foundation and all the research that were occurring in these different areas of the world. And so we could measure the distribution of secretors and non-secretors throughout the world. And we can also then sort of profile or provide the profile of mothers in their milk who are non-secretors versus those who are secretors. And so we extended the study. So we not only human milk oligosaccharides, but we also looked at milk oligosaccharides from other mammals. And so we looked at, of course, cows. We've been trying to get an echidna ever since, but never did. We did get some platypus. We've done kangaroo. We've also done pigs. More recently, we did cats and dogs, and so that was an interesting. The data there was really interesting because dogs and cats actually, it seems like they represented one secretor phenotype. Dogs were more like secretors and cats were more like non-secretors. And the other bizarre thing there is that dogs and cats actually have oligosaccharides closer to humans, even closer than some primates. But in general, primates were a lot closer to humans, as you would expect. So the last part of this of this chapter to us in HMO analysis was that could we develop a method for quantitation going from relative to absolute abundances? So from the relative abundances, we can get that from the area under the curve. However, there are we lack standards for doing absolute quantitation. And so the method that we came up with is to sort of interpolate what the abundance would be based on the structure. So for example, we may have this standard, but we don't have this standard. And so we wanted to know what the effect of a positional isomers are on the actual abundance itself, on the signal response. We may also have a structure that looks like this, but then it would be extended by a sialic acid. And so we wanted to know what that would do. And we may have a standard that looks like this, but then we would have that more fucosylated versions of this. And so there we wanted to know what those would do. And so we did that. We systematically looked at the standards versus those that are between those different changes and looked at their response. And then the other thing that we did was we actually automated the whole process. And so now we could do this on a 96 well plate. We do the centrifugation and the drying, the shaking. We do that using this device, which is an Opentron. And so we can do now many samples quite reproducibly. So one of the things that we did was we created a standard pool after we figured out all the response of each of the different oligosaccharides. And then we again switched to a new type of mass analyzer, this time where it was more a targeted method. And we started to then do absolute quantitation using the standards and the new standards that we created using the method of interpolation and extrapolation of the different structures. So based on 15 standards that we had that we could purchase and we can verify their purity, we then determined the absolute abundance of some 60 more or 60 total human milk oligosaccharides. And this is just to show you what the response is and how the performance are when we start using the extrapolation. So in general, we get about an 8. So we created a validation set of structures and we tested those. And so the difference between what we would predict and the actual is about 8%, which is pretty good. In some cases, it would be as high as 25%. But again, we're quantitating structures that you can't normally quantitate because there would be no standards. And so, but this is a bigger problem, not just in human milk oligosaccharides, but also in the metabolomics community and other places where it's impossible to get the kinds of standards that you would need to quantitate dozens, scores, or even hundreds of different structures. So, in any case, this is a format that, and this is, by the way, work by Aaron Stacy, who single-handedly developed this last part. And so what he did was he used this format to represent the different concentrations. This is 2-fucosyl-lactose, it's in yellow. This tells you, and each of these are the different structures or the different abundances of these different compounds, the different HMOs. But what you could see here is that this is a secretor because it has a lot of 2-fucosyl-lactose. And these are the structures that are present here. And this is a non-secretor because we don't have the big yellow. But these are all the different structures that are contained in these different in these tables, or at least in the different absolute abundances in this format. And so you can actually you can pre-map these are different mothers and their different compositions of milk. And so even by looking at this, you could see that these are secretors, these are non-secretors. And so this sort of makes organization a little bit easier, but it also shows you the variation between the different mothers and their human milk oligosaccharides. And so with that, I'm going to conclude. So my conclusion is that, again, to profile the human milk oligosaccharides, we've developed these new technologies. We created a high-performance MS analyzer for ultra-high resolution mass and mass accuracy. We developed new chromatographic methods to separate oligosaccharides, also in standard and nano-LC format. And we created methods that can annotate the structure and identify structures de novo and even identify them rapidly. And we developed methods for absolute quantitation, both relative and absolute quantitation. This work has been over the past 15 years. It's resulted in over maybe about 200, 150, 200 publications. It's graduated dozens and dozens of graduate students. I can't name them all here, but recently at the American Society for Mass Spec, we had an alumni social gathering, and these were some of the people who attended the meeting. And I think the earliest one here was from 2007, who graduated from my lab in 2007. But in any case, many of those were involved in this project, and this is our funding agency. And I'd like to thank you all for your attention, and hopefully this was useful for you.
Wow. Thank you, Carlito. That is such a magnificent demonstration from beginning where structures were not possible to be elucidated to now Carlito has brought precision medicine to the field of maternal lactation all through the work that they've been doing. Just magnificent. All right. Going to be tough to top that, but the next speaker is one of the great friends I have in science. I've known Lindsay Allen most of my scientific career. She is a hero of science. Don't ask me. Look at her CV. Her bio sketch is just a testament to heroic work on understanding lactation, babies, and how milk can be the basis of understanding how nourishment works, full stop. The next phase is understanding milk in its entirety. Instead of a reductionist approach, looking at one thing at a time, build a basis, a database that acts to look at all things and build quantitative assessments of milk. That's a global initiative. There's only one person who can achieve that. Don't ask me. Ask the Bill and Melinda Gates Foundation, the best person in the world to take that on, Lindsay Allen. And Lindsay is continuing to make a massive impact on the field of milk and infant health, and we're delighted to have her here. Lindsay.
Yes, thank you very much. Okay, welcome everybody to this really interesting short series on human milk and particularly human milk composition. Since human milk is so critical for infant nutrition, it's obviously important to understand its composition. So there's been a lot of work on this in recent years, as evidenced by the other presentations today and the literature. You might think we know a lot about the amounts of the basic nutrients like vitamins and minerals in human milk. But until the study that I'm going to describe today, we really did not. The limitations are sample collection and analysis, in other words, diagnosis of what's in the milk. And this presentation is with Daniela Hampel, who is the chemistry whiz behind this, and she will be on the panel later. This work is supported by the Gates Foundation. Here are disclosures on this slide, and none of this work on my part on the right is paid input. So next slide. Can you see the background?
Yes.
Okay, fine. So as an introduction to this talk, it's really important to know the nutrient intakes of infants and their output by mothers. Currently, the recommended nutrient intakes for infants are based on published data on the concentrations of nutrients in human milk. The problem is there are very sparse data on milk composition, especially the amounts of vitamins and minerals in the milk. We found out as we started this research and collaborated with many people around the world that the amount of micronutrients, vitamins and minerals in human milk are very low in some population groups. But we didn't know the answer to what levels are really low. We needed some reference values to compare them against. Unfortunately, the global status of nutrients in human milk is almost unknown, as of course then subsequently the physiological effects of deficiency, the effects on the infants, and then the relationship with other micronutrients in milk, other milk constituents and the micronutrient and the microbiome and so on. As Bruce said, we need to start looking at milk as a whole package, which the constituents of which relate to each other. So this study is about developing reference values for milk nutrients, and we're also doing maternal and infant status biomarkers in order to understand this situation. Next slide. So we were asked to name 3 challenges and solutions to doing this diagnostic project. And so the first challenge that we had was how to collect and analyze milk samples. And we did this in what's called the Mothers, Infants, and Lactation Quality Study. We call it the MILK study. And this work was done primarily with USDA in Davis, the Western Human Nutrition Research Center, but also with 5 wonderful collaborators in Denmark, Dr. Kim Michelson and Christian Molgaard. In the UK, Sophie Moore, who was working in a well-nourished area of the Gambia. In Brazil, Gilberto Cac at the University of Rio de Janeiro. And in Bangladesh, Dr. Munir Islam at ICDDRB. And the design that we chose was to have well-nourished but non-supplemented mothers. You couldn't do this in the United States because women take supplements in variable ways at variable times, and this definitely affects the amount that's in milk. And what we want is like a reference value, what is, quote, normal. So in these 4 countries, we had 1,000 mother-infant dyads, and each of them had 5 visits between birth and 8.5 months postpartum. Lots of supporting data, including plasma status biomarkers and diets of the mothers and infants, microbiome, metabolomics, HMOs run by Dr. Labria's lab, genomics, and so on. So the first solution had to be how to collect the milk samples. And this is what we call the breast milk quality study. So this design took place in Bangladesh, and we had 3 days of complete milk samples from mothers in the study. And day 1, it was the baseline where we gave no supplements. We just collected milk, no supplements, no food. Day 2, we gave 1 supplement, the RDA, in the morning. And day 3, we gave twice the RDA. And so every feed for 24 hours every day, we collected 4 milk. That's the first 2 minutes, the rest of the milk, and then a full breast expression, which is the sum, obviously, of the first 2 collections. And what we learned from this was that, as you can see in the accompanying graph, that there really wasn't much difference between the 3 samples, among the 3 samples, for any of the nutrients. And so circadian variance, collection time during the day, was only a minor contributed only 7% to the total daily variance. The main variance was between women, not during the day. So there was also no major difference between the 2-minute samples and complete breast collections. And then we did a follow-up study that looked at the effects of acute supplementation. And so the 1 RDA and 2 RDAs, the bars here are day 1, baseline 100%, day 2, 1 RDA, day 3, 2 RDAs. And you can see great variability in the nutrients in terms of what responds during the day, particularly riboflavin, the second set of bars, B6, and so on. And so the lesson here was that you cannot collect milk soon after a supplement or a meal, because that would affect the levels of nutrients in there. But likewise, if you just collect baseline, you just might miss the effect of supplements on milk. So this is a very tricky thing to figure out how to deal with this.
I'm sorry, but we need to advance to the next slide. This is the supplementation slide right now.
Okay. Okay, so everybody see the large increases in riboflavin and B6 as a result of supplementation in the morning.
So now this is the where you see the increase due to that acute supplementation, where you see the B2 and B6 rate increase. And I'm going to move forward now. Hold on. Here we go.
Great. Okay, so the other very difficult aspect of this, which I have to say took 10, 12 years to figure out, thanks to Daniela, is how to analyze the milk samples. Milk is very complicated as far as the nutrients in it. There are many vitamins. The left-hand box here shows you, for example, it's not just thiamine, the thiamine monophosphate, pyrophosphate, and so on. So there are vitamins that we have to analyze as well. So the real workhorse here was UHPLC mass spec, and we published many papers on the methods, or Daniela has done most of that. And I think this has been a major contribution to milk science. B12 is a immunoprotein assay. We did some cytokines and hormones with multiplex. The other mass spec was an ICP mass spec in which we analyzed about a dozen minerals. A lifesaver is a near infrared spectrophotometer, which does protein, fat, carbohydrates, and energy. And then more or less usual methods for vitamins A and E and carotenoids using HPLC. Next slide. Okay, so the other next challenge, number 2, is the study design. How are we going to collect human milk to be able to say, these are reference values? And we wanted to look across lactation because it was well known that particularly in the first months, there are major changes in the vitamins and minerals in human milk. So there was early recruitment antenatally, and the criteria for the study were that the women were all well nourished and they would deliver colostrum. We had what's called an early milk project, E1 and E2 during the first months, which was added later because we were worried that the women would give up if we loaded too much of them at once. And then there was milk 1, 2, and 3 periods with rolling enrollment during those periods. So the same woman would enter, say, early in M1, early in M2, early in M3, and another one would enter later. So we could have data for almost every day of these 8.5 months. So the protocols had detailed anthropometry and breast milk volume that I'll mention more in a moment, morbidity and lactation questionnaires, and the milk samples, of course. Next slide.
Ready?
Yeah. And then we had to express the data over time. And this was thanks to our wonderful statistician, Janet Pearson. And these are just a snapshot of some of the data. So on the left, you see the country codes. And in the middle, we see, starting with thiamine, vitamin B1, changes for the whole group, 50th percentiles, and the actual data points across the first 8.5 months of lactation. The second graph on the right shows the percentiles going from the 5th to the 95th percentile, and the data are available as percentiles across the whole spectrum of values. And then there's riboflavin, and you can see for riboflavin, here's niacin, big increases in the first months and then fairly flat after that. B6 increases during the first 2 or 3 months, and B12 at the bottom is mostly flat except for these weird results from the Gambia where the mothers and the babies have very high levels of B12 reflected in milk as well during the first couple of months for reasons we don't understand yet. Another thing you can see from these graphs is that in some of them, for example, the B12 at the bottom right, that Bangladesh has lower values than the other countries. Gambia has higher ones all the way through the period. Next slide.
Ready?
Okay. So another novel aspect of this was that we measured milk intake across the whole spectrum 3 times in each mother-infant pair at the same time or in the same short period that we were collecting the milk to analyze the micronutrients. So we couldn't answer, for example, or nobody I found could answer, if a mother who typically produces more milk has more micronutrients she's delivering the baby, or the micronutrients are just more dilute in the sample. Well, what we found was that milk volume was only weakly correlated with nutrient concentrations. So basically, that means that milk volume, the typical amount consumed by an infant, and it varies enormously among mothers, was the main predictor of infant nutrient intakes, something that's really not taken into consideration very often. Next slide.
Okay. Ready?
Yep. Okay. The third challenge, after we'd realized how to collect milk and express the data, was to apply the results to improve nutrient intake recommendations. And so nutrient requirements, which are called adequate intakes because the data are so poor at the moment, that's enough sort of approach. Those are based on estimates of milk concentrations of nutrients, which are very poor in the literature. Some of them, for example, riboflavin, is based on data from only 5 women 25 years ago. And the other thing is actual intake, which is volume. We measured it. Usually it's just assumed to be 780 milliliters per day for everybody, which of course is not the truth. So we now can use the total intake values from the milk study to set estimated average requirements and recommended dietary allowances, which is what we need to be able to say that the infant is getting enough or how many are not getting enough and so on. And so the table on the right shows you the values that we got from the milk study in terms of concentration, from the Institute of Medicine, which is about the only global agency, well, it's not global, the only agency that's making recommended intakes for infants. And finally, the milk study is a percent of the Institute of Medicine. So in general, the values we found were lower than the estimates by the Institute of Medicine. A few were higher, some were very different. So what we're hoping is that global agencies and the IOM will use these values to develop better estimates of intake by nutrients and intakes requirements by lactating mothers and the infants. Next slide.
Okay.
The other main use of these data is a diagnostic one. We can assess effectiveness of intervention programs and supplementation by looking now quantitatively at the change in milk and whether or not the impact is to move values above the median or from very low values to the median and so on. This is a study within the milk study on universal salt iodization and its impact on milk iodine. And you can see that the milk iodine is much higher in Bangladesh due to their programs. Every country had different levels of salt iodization, but you can see the impact there. We also had a lot of thyroid metabolism measurements to be able to evaluate whether these milk values really related to the iodine status of the infant and the mother. And the next slide.
It's up.
So here's vitamin D as another example. And you can see the very low values in Bangladesh. The mothers were frankly deficient. The milk is very low. And this was sort of generally not realized that there was this difference in milk. The need evidently for a fortification or supplementation program. Denmark in red was perfectly normal and in tune with everybody else, even though D deficiency is a risk in Denmark, and that is solved by the mothers receiving adequate supplementation. Next slide.
Ready.
Okay. Yep. So this is the last example of application, and this is a preliminary work that we're doing. We are looking at the change in the percentiles in human milk as a result of supplementation. So across the bottom are some of the studies we've been involved in. The green marks are the control groups, and the orange ones are the supplemented. Now I have that backwards, sorry. The green is supplemented, orange are the control. And so what you can see from this is that some countries, some sites have very low values to start with, and the supplementation increases the values. Others, interestingly, in which there was a huge effort at supplementation, were fine to start with, only they didn't know it. And so this is a way now we can start to look at human milk composition around the world in terms of adequate percentiles. Next slide. So what we're going to do now, future directions for the milk community. We want to use these data to model predictors of milk nutrient concentrations and volume, using everything such as dietary data and genetics and the microbiome and so on. We want to involve agencies to improve intake recommendations, to use the milk nutrient concentrations, hopefully as a tool for nutritional status assessment of the mothers. And we want to use them to guide whether supplementation or fortification is needed, the timing and the dose and so on. And we'd like to look at, or we'd like others to look at, milk quality across populations. For example, there's a national survey of nutrition going on. Collect milk and see if it relates to other biomarkers, because you can analyze so many nutrients at once, which you can't really do very easily with blood. And then finally, we'd like to know how these percentiles link to functional outcomes in the infant. And the last slide. So from the milk study, we learned how and when to sample human milk and how to analyze the micronutrients in human milk. And nobody before had put together this whole package. Infants' nutrient intake is more strongly predicted by the amount that they consume than the concentrations in milk. So that should always be remembered. We can estimate or update the current adequate intakes for infants to become recommended intakes. If we had better intake recommendations, this would improve our estimates of nutrient gaps between milk intake and what should be formulated in complementary food, composition of formulas, and so on. And then finally, milk micronutrients might be the best tool to assess maternal micronutrient status and the need for and effectiveness of maternal supplementation and food fortification. And I'd like to thank our funder, the Gates Foundation, who's hung in for about 12 years to develop all of these data, and the very excellent collaborators and field workers and the lab workers in this study. Thank you.
Oh, Lindsay, magnificent. That is truly heroic. And it's amazing for me to see how much impact a single driven individual can make on behalf of babies everywhere. Thank you, Lindsay. Now we're going to turn to Nicole Kochko. And it is, I think, one of the great success stories of the history of science that the research clinician has driven science both ways, demanding of scientists to study the critical priorities, what do we need to know, and then bringing knowledge to practice. There are so many things you have to do to be able to be a clinical researcher. They are rare. And they're even rarer who address milk, lactation, and babies. And the rarest of them all are those who work on behalf of premature babies. And Nicole is one of the young stars of this field. We are delighted to have Nicole here.
All right. Hi, and thank you for this opportunity and invitation to speak today. Like Bruce said, I'm Nicole Cacho. I'm a neonatologist here at UC Davis, and I will be discussing enteral feeding practices and nutritional challenges for preterm infants in the NICU. I have no disclosures to make, and our learning objectives for today will include discussing donor milk variability, talking about the 3 nutritional challenges and solutions to those 3 challenges, and also future directions for neonatal nutritional research. There are 15 million preterm births every year. Mother's own milk is the best base milk for these preemies. It provides protection against infections and it reduces the risk of necrotizing enterocolitis. NICU mothers face many barriers, including delayed secretory activation, low milk supply, and separation from their infants. NICU-specific interventions include many, but the main ones we focus on are early expression, ensuring double hospital-grade electric pump availability to the mothers, lactation support specific to the NICU, and facilitating frequent pumping, at least 5 times per day. Necrotizing enterocolitis is a serious gastrointestinal illness in which the tissue lining of the intestines become inflamed and die off. NEC is one of the leading causes of illness and death in preterm infants, particularly those born with a very low birth weight. Unfortunately, 1 infant dies almost every day from NEC in the United States. The only thing that has been found to be protective against NEC is human milk. Survivors of the most severe NEC are at a significantly higher risk of lifelong neurodevelopmental problems compared to those preemies who do not develop NEC. When a preterm baby is born, a mother is not always able or willing to produce adequate amounts of milk to nourish her baby. In these situations of low milk supply, many consider donor milk as the next best option, preferred over formula. The main reason we use donor human milk in the NICU is for the decreased risk of NEC when mom's milk is unavailable. Donor milk is offered as a bridge to mom's own milk, but many infants receive it for more than half of their NICU stay. Though donor milk offers protection against NEC, it remains nutritionally inferior to preterm mom's own milk. Again, donor milk's role is to be a bridge to mom's own milk. Most donor milk is from mothers of term infants who are at least 6 months old. This milk is a very different substrate compared to early preterm mom's own milk. Furthermore, processing and storage decreases bioactive components and eliminates enzymes. In the United States, it is standard practice to add fortifier to human milk fed to preterm infants. The reason we add fortifier is to increase caloric density, protein, and minerals to match preterm infants' increased demands. The use of human milk fortifiers varies greatly between each NICU, even NICUs in the same city. The human milk-based diet often fluctuates in percentages of each different milk type during hospitalization, yet it's treated the same. So on to our first challenge. Donor human milk contains lower sodium than earlier stage of lactation, preterm milk. Because preterm infants waste salt, they are at high risk for low total body sodium. This risk is exacerbated by multiple medications we use for preterm infants in the NICU, especially diuretics. The result of low sodium for these babies is unfortunately poor growth. The most common way we detect hyponatremia in the NICU is noting low serum sodium on routine labs. Beyond poor growth, emerging research suggests that low sodium is associated with sepsis and even neck. Additionally, low sodium can also affect protein utilization. Since sodium can affect so many important clinical outcomes, we need to consider actively testing the milk enteral levels instead of passively detecting low serum levels. An innovative approach is to consider the use of point-of-care sodium milk testing at the bedside. Point-of-care sodium testing is already being used for detecting timing of secretory activation in some NICUs and in the management of mastitis. Instead of a one-size-fits-all fortification, we can measure sodium milk levels at routine time points or when we know there's a higher usage of donor milk in certain babies, and then add targeted sodium supplementation. This personalized approach, counting the base milk plus the targeted supplements plus the appropriate human milk fortifier, could transform how we feed our most vulnerable infants. If point-of-care sodium appears promising, it will need to be validated for both mom's own milk and donor milk, and also have standard target ranges. These ranges may need to mirror the stage of maturity of the infant to match milk's changes with corrected gestational age, like you would see in mother's own milk of a mother that is pumping and providing all of her milk for the baby over time. Also, cost-effectiveness analysis should be performed. So on to our next challenge. The protein problem. It is well accepted that early protein delivery supports growth and neurodevelopment. Donor human milk has lower protein than preterm mom's own milk, but the protein level varies, especially based on processing practices. Insufficient protein delivery leads to poor growth. Process variations include number of pooled donors, stage of lactation, prematurity of the donor's infant at birth, and if target pooling is done with macronutrient analysis. such as using a human milk analyzer. To illustrate the effect of some of these processing practices of donor human milk, we will review two different figures. First, let's focus on the figure on the left. This is from a study published in 2020, looking at the number of donors in each pool. As you can see on the x-axis, the number of donors start at 1 and goes up to 5. Protein level is on the y-axis. As the number of donors increased in the pool, the variability in the protein content also decreased, creating a more consistent, reliable product. Now let's look at the figure on the right. This is a separate study published in 2023, which looked at the stage of lactation of some preterm infants in purple and term infants in teal. It showed that the stage of lactation strongly impacted the protein level. In our unit, we are conducting the Preterm Infant Nutrition and Growth Evaluating Donor Milk Impact Study, also known as the PING study. This is a retrospective observational study to evaluate infant growth during 3 epochs of donor milk use in our NICU. The first epoch started when we initially began using donor human milk from milk bank number 1. Anecdotally, there was worse growth after implementation of donor milk. So we started fortifying straight to 26 kilocalorie per ounce, and this marked the second epoch. During the second epoch, my colleague purchased a Mirus human milk analyzer with her grant award and found the milk from milk bank number 1 to have a protein of only 0.6 grams per deciliter when it was presumed to contain 1 gram per deciliter. This was a big difference. Concurrently, milk bank number 2 started offering high protein donor milk measuring between 1.2 and 1.4 grams per deciliter. Our unit decided to switch to the newer high protein donor milk, and this marked the third epoch. Anecdotally, as time moved on, growth has gotten better, but this study is IRB approved and it's ongoing. So our PING study highlights the importance of quality control to verify protein content. Our unit's experience also demonstrates how processing techniques can be utilized to produce high-quality needed products, such as high-protein donor human milk. This change on the milk bank side eliminated the need for us to fortify straight to 26 kilocalories per ounce. Milk bank practices can be optimized to reduce variability, and this is what Dr. Chan will be talking about today. But individualized analysis and fortification may be needed for precise nutrition in some cases. In many clinical scenarios, HMF protocols may need to be adjusted based on system or practice changes, or even personnel availability. Comparing all milk bank processes and current product output will lay the foundation for meaningful research, including the ability to track long-term growth and development. So our last challenge of the day, fortification. Since protein is so important for growth and development, it's the main clinical focus when we talk about fortification in the NICU. Yet many questions remain. First of all, what's the right quantity of protein? How much do you give babies? Protein goals are based on expert opinion using fetal accretion rates as a reference. Also, is there an upper limit that can lead to overnutrition? This is an important counter argument to consider in the setting of increasing obesity and cardiovascular disease in adults as well as children. Next, should we provide powder or liquid? We need to think about safety of powder and volume displacement issues with liquid fortifier. And as Dr. Allen taught us, volume consumed is very important. So liquid fortifiers do displace the base milk of donor human milk or mom's own milk. So that's a consideration for liquid fortifiers. And lastly, consider the source. That is, what's your protein source? Human versus bovine, whey versus casein, and also amino acid based versus peptide based fortifiers based on the degree of hydrolysis. There are limited studies examining long-term outcomes in human milk fortifiers. And surprisingly, there's little known about metabolic programming effects. Human milk fortifiers are difficult to study due to the variability in product use and even current protocols using the same fortifiers. Here is some preliminary data from the lab that I work in, and this is on fortified donor human milk using 2 different bovine fortifiers that are commonly used in the United States, and their amino acid profiles. Although all essential amino acids are present in each of the different fortifiers, the amino acid profiles of the HMFs were distinct. These unique amino acid profiles led our group to continue to focus on newer clinical outcomes of these fortifiers, such as implications for metabolic dysfunction. Let's start by looking on the left at figure A. This is a dot plot of the difference in the 2 most commonly used bovine human milk fortifiers in NICUs. The red represents the amino acids that are higher in the partially hydrolyzed fortifier, HMF1, and the green dots represent the amino acids that are higher in extensively hydrolyzed fortifier, HMF2, which is the product we actually use in our NICU at UC Davis. Starting with figure A, we can focus on tryptophan, the 3rd amino acid from the top. It's also the 2nd red dot from the top. It's an important amino acid, rich in the whey protein, and it's a signaling molecule and precursor to serotonin and melatonin, and thus important in brain development. And we can see this is higher on the fortifier on the left. Now let's look at figure B on the right. This is a heat map of the amino acids in the same 2 bovine fortifiers. Orange means that the amino acid is mainly from the fortifier, and blue means that the amino acid is mainly from the donor milk, which was the milk base in these mixtures. The amino acid profiles are vastly different between these 2 bovine fortifiers, as evidenced by the various colors and intensities. Again, we will focus on tryptophan, the 3rd amino acid from the top, and we see more tryptophan is provided by the fortifier on the left, the HMF1, as it's darker in orange intensity. In summary of our 3rd challenge of fortification in the US, it's standard practice, but there are 3 main products which are distinct in most protein categories. Whether focusing on protein quantity, protein source, or the degree of hydrolysis, or even the carbohydrate source, there is variation in all of these characteristics of all the fortifiers we use in the United States. If we standardize our approach to evaluating the nutritional substrate, such as the base milk, the fortification, and supplements, such as sodium supplements, continued research on meaningful outcomes such as necrotizing enterocolitis, neurodevelopment, and metabolic dysfunction is possible. We also need to partner with families to get their input on which of these outcomes are important to them. So, in a review of the 3 challenges I presented today, please remember, mother's own milk is the gold standard, but many infants will still use donor human milk. So we need to optimize donor human milk when mom's own milk is unavailable. Current standards include targeted supplements and fortification of donor human milk. But also remember, donor human milk has lower sodium and protein compared to preterm mom's own milk. A thoughtful approach is required to consider which human milk fortifier to use for fortification. The time has come for standardized protocols across NICUs. Still, a personalized nutrition approach is warranted based on growth trajectories in specific cases for each of our babies. Team science will require industry, basic scientists, and physician scientists to work together and form strong partnerships for innovation and sustainability. Also, we cannot forget about advocacy and policy change. This must begin and continue to support parental leave, on-site daycares, lactation, and equitable donor human milk access. New partnerships should be formed with neonatal nutrition focus, such as the creation of a brand new first Society of Neonatal Nutrition and Lifelong Health, which was just created within the past week. And I'm honored to be a part of this. I have a lot to be thankful for, and here are my teams that keep inspiring and supporting me.
Nicole, magnificent. Premature babies are in such good hands. I have so many questions. We cannot sort the questions yet, but I look forward to asking you questions later. Thanks, Nicole. And now I am absolutely delighted to welcome a fellow Canadian, Professor Tim Chan from the University of Toronto. I encourage you to read his biography if you have a lot of time. Tim is a true star, celebrity in Canadian science. I'm jealous. Tim's knowledge of his field is so broad and so deep, he is pursued by all of the important media. No medium is more important in Canada than the Canadian Broadcasting Corporation, CBC. Tim has been a resource for CBC multiple times. As a 10-year-old child, I was on the CBC on behalf of an ice hockey team, and I've never been asked back. I am so jealous of Tim. Tim is going to talk to us about the new tools and new capabilities that mathematics is bringing to the world and especially science. It is time for the modern computational universe to embrace human milk. Nobody's better to help us with that than Tim. Please, Tim.
Thank you, Bruce. That was far too generous of an introduction. And for the record, I've never been asked on CBC for hockey, so you're one ahead of me there. Okay, so first of all, I want to thank Bruce and Jenny for this very kind invitation to present at this ROS workshop. This work that I'm presenting today is a project that was done that spanned a couple of years in collaboration with several students and colleagues from the Milk Bank here in Toronto and at the University of Toronto. And I have no conflicts to declare. Okay, so Nicole I couldn't think of a better kind of introduction to this work and providing so much important data and motivation for the work that I will be presenting here around milk banks. So for those of you who, so I assume many of you here know what a milk bank is and what a milk bank does. And for those of you who don't know, I'll just show kind of a very brief video that describes a patient who benefited from the milk of the milk bank here in Toronto to give you a sense of the type of patient that this milk bank serves.
27 weeks, Thomas was born, and shortly at that time, I was diagnosed with stage 4 cancer in my liver. I started chemotherapy, and I wasn't able to produce milk. And so when Thomas was born, it was a very easy decision to give him the best start we could by using the donor milk program. We wanted him to have the best chance that he could possibly have, and having done lots of reading and research, we knew that breast milk was the way to go. And because I was ill, I was not able to make any at all. And so Thomas was 100% fed off the donor milk program. Oh, he's doing really well. The doctors are very happy with his growth. He's at 37 weeks, almost 37 weeks today. And so he's almost at term. When Thomas was born, he was 1 pound, 13 ounces, and he's now 5 pounds. So he's done really, really well.
Okay, so I think you get the idea here. So, as was mentioned, mother's own milk is the gold standard. But if you're sick, in this case, like Sarah, and undergoing chemotherapy, she was not able to produce milk, and so her baby was fed from the donor milk program. So just a little bit more background on milk banks. So milk banks, they collect, they process, and then they distribute this milk to NICUs or potentially to the community even. This picture on the top right shows the locations of nonprofit milk banks in North America. One of the key steps that is in the process of making the milk or pooling the milk in the milk bank is this kind of pooling process, where depending on the milk bank, there will be different guidelines or processes for how you do this, but it typically involves pooling donations together from multiple donors. And so I think, as was already explained very nicely by Nicole, one of the key features of doing so is actually reducing variability. She showed it in protein, but I think it's true in general for nutrients beyond protein as well. It also increases the, excuse me, the variety of bioactive components in the milk. In terms of the pooling step, I think there was a couple of pictures of milk analyzers in the previous presentations as well. And the presence or absence of an analyzer will dictate pretty significantly the pooling practice of a milk bank. Not all milk banks have them. Not all milk banks are approved to use them for this purpose. And actually, over half of nonprofit milk banks in North America either do not have one or do not use one for this purpose. So as a result, if you don't have, think about all of the challenges that were described previously, and maybe coming up first and foremost, one major challenge is, what if you don't even know what's in the milk? Okay, so you want to pool milk together to make a high quality milk product, but you actually don't have a good idea of the level of protein in the milk, the level of fat in the milk, and so on. And then so in that case, what do you do? One option is to use heuristic strategies for how to pool the milk. So FIFO stands for first in, first out. So milk has an expiration. Maybe one idea would be to basically pool milk together that is, that is the oldest in the sense. So the ones that are going to expire the soonest, so that you minimize wasting milk, as an example. Does that produce the best quality milk in terms of nutrition? Probably unlikely, but that is one thing we can do if we don't know what's in the individual donations. So what did we do in this project? So this was a collaboration between kind of engineering optimization people like myself. We had a neonatologist on the team, a nutrition scientist, a nurse who was the director of the milk bank as well. And so we collectively came up with this framework for donor milk pooling using optimization. The motivating goal is really to help nonprofit milk banks that don't have an analyzer or aren't approved to use one for this purpose to optimize the quality and consistency of their milk. And so there's really two key challenges I'm going to talk about today. One is what I've already alluded to, is that if you don't have an analyzer, how will you know what the macronutrient values in each donor deposit are? And then the second challenge is, even if you did know what was in the milk, how would we best combine the deposits or pool the deposits in order to make a consistent and high quality milk product? So to give you an idea of what the current approach looks like in a schematic, this is a picture of one of the whiteboards in the milk bank, and it collects very high level information. There's other information that's recorded as well. But to give you a sense, a lactation consultant will look at this whiteboard and then make this recipe on the right-hand side. So they'll know things like the date of birth of the baby of the donor mom who's donating, whether or not this is a one-time donation or an ongoing donation, kind of the date range of the donation. We'll have some information about bacterial colony counts. We'll have some information about how big the donation is in terms of liters of milk. And then from this fairly high-level information, a human has to make a decision of saying, on this day, I'm going to pool this amount of milk from these 5 donors, for example, in order to make my recipe for today. Okay. And our goal is to, in a sense, replace the human with some sort of AI, some system that actually can infer or know more potentially about what's in the milk to make smarter decisions around how to pool the milk and namely what quantities from which donors to pool together. Okay, so before I tell you about what we did, I will just briefly review the process for those of you who may not be as familiar with a milk bank. So the first step is screening and approving donors. So there is an online form. This is, again, the process at our milk bank, which may vary from milk bank to milk bank, but hopefully the overall process will be somewhat similar. So there'll be some process for screening and approving donors, doing some labs, and so on, collecting some information from the donors. Once a donor is approved to donate, our milk bank will send empty boxes, these pictured there. They will be insulated on the inside, and there will be empty bags or containers for the mom to pump milk and then fill these containers or bags, store them in her freezer at home until 5 liters, or I believe it's 5 liters, is accumulated or more. Once a certain amount of milk is accumulated, they will put the milk in the box, ship it overnight to the milk bank. The milk bank pays for all of the costs of this process. Once the milk arrives in the milk bank, it'll be cataloged. It'll be logged into inventory. The staff will start then a series of tests for bacteria in the milk. And then the milk will be stored in this freezer on the left-hand side, and then until it's ready to be used. And then the night before a particular donation will be used, it will be put into the fridge on the right-hand side to defrost. And the day, or typically a few days before, potentially up to a week before that day of creating the recipe, pooling the milk, the recipe will be created. So this is that whiteboard picture that I showed you on the left. And from the whiteboard, the staff at the milk bank will create a recipe that looks something like this, that says, we're going to take 5 liters from donor 1, we're going to take 5.9 liters from donor 2, 5.7 from donor 3, and so on. And then once that recipe is created and the milk is thawed overnight, that next morning, the diet techs will, as you see in the picture on the left, will take the milk, will distribute it to various beakers according to that piece of paper that's stuck on the wall there you see there. And then we'll do another test of bacteria from the pooled milk this time. And then the milk will be on the left, you see homogenized and mixed on that mixer, and then aliquoted into these bottles that are then heat sealed using the machine on the right, and then placed in that wire rack that you see on the right-hand side there. And then these wire racks will then go into a pasteurizer. So here at the milk bank in Toronto, it's thermal pasteurization, so with heat. And then, the milk will be labeled, and then it will go into a different freezer for storage until the milk is shipped off to a NICU that is looking for milk. Okay, so now that you have a sense of the process, maybe I'll just go back to say that we're not changing anything in this process other than this step right here. So our idea was to simply come up with a model to then recommend the recipe for the day or for the next few days, and then everything up till this point in the process and everything after this point in the process remains unchanged, so that it can be hopefully fairly easily and feasibly incorporated into the current workflow. So now I'll come back to the couple of challenges that I mentioned earlier. So the first challenge is around the unknown macronutrient values in the deposits. And so here, our solution was to use machine learning. And so we're focusing specifically on fat and protein only in this study as a proof of concept. So the idea is, could we predict the amount of fat and the amount of protein in a donation based on, without using an analyzer, but based on readily observable data from the patients, or from the donors, like the age, like the dates of donations, like the volumes of the donation. Okay. And so the idea is, just in a schematic form, we would take donor and deposit variables, or donor and deposit data, or covariates. We would input it into a machine learning model to be able to predict fat and protein. Now, of course, an analyzer was used in training the model, because we needed to give the model historical data of pairs of, let's say, donor deposit variables and fat or protein values. So the analyzer was used from a research perspective to train the model, but once the model was trained, then we don't need the analyzer anymore. We can then just feed it, unseen donor and deposit variables, and then it will make a prediction for what the fat and protein will be. Okay, and then so the second challenge is around combining deposits together. So here, we developed what's known as an optimization model, and the optimization model determines the volume of different donations to pool together. And it has to account for various operating constraints that the milk bank operates under. So, for example, it operates under a certain kind of bacteria threshold, a pre-pasteurization, even when I'm not talking about BC post-pasteurization, but even pre-pasteurization, they want the milk to be below a certain colony count. So the optimization model has to decide which donations to pool together to keep that overall bacteria level below the threshold. We have to worry about things like how much capacity is in the pasteurizer to pasteurize every day. We have to worry about workload considerations for the staff at the milk bank. We have to worry about the milk expiration dates and so on. So the model takes all of this into account, and it spits out for us this recipe, one per day, to say, these are the 3 or 4 or 5 donors that we are going to pool together on this day. And again, so the kind of visual to think about it is, the optimization model needs to get estimates of fat and protein, and it comes into the optimization model, and then the optimization model knows everything else around the bacteria, the pasteurizer capacity, and so on, and then it will output this pooling recipe. So how does this all fit together? If you only remember one slide from my presentation, I will just ask you to try and remember this one, okay? And so here's where we take our two solutions and we stitch them together. So we start with the basic data that the milk bank is already collecting around the donors and the deposits. It goes into a machine learning model that predicts the fat and protein for every donation. And then, given those predicted values of fat and protein and other variables, it goes into the optimization model to then produce the recipe. Okay. And the way we implemented this is we did it on a rolling 1-week basis. So let's say today I will make the next 7 days or 6 or 7 days worth of recipes, and then I will implement those over the next 7 days. And then a week from now, I will look at what milk is sitting in inventory at that point, and then make the next 7 days of recipes, and then so on. Okay, and so the last thing I'll describe is our implementation. So we did a trial implementation at the milk bank in Toronto. So it's a mid-sized nonprofit milk bank, one of about 40 in North America. It services 51 NICUs in Ontario and the Maritimes, which I've been told is maybe a foreign phrase for people outside of Canada. So that basically means the provinces kind of out east. This milk bank processes about 10,000 liters of milk a year that come from roughly 800 new donors. And some returning donors, obviously. And an important point to emphasize is that this trial was implemented during regular operating conditions of the milk bank. So it was not a controlled experiment. The milk bank was just kind of operating like normal. We didn't influence any other aspect of the operation other than basically switch out who was making the recipe. Okay. And so here's the timeline for our study. So we started back in the fall of 2021. And for about 6 months, we had what we call a baseline period. So in those 6 months, all we did was we observed the milk bank operating, excuse me, and we simply recorded metrics like what were the fat levels in the milk being produced, what were the protein levels. There were certain target fat and protein thresholds that they would like to meet, and so we would measure what fraction of the pools actually met those targets. We would measure things like how many batches failed due to BC, how many batches failed due to bacteria, pre-pasteurization, variability in the protein and fat over time, and so on. So we basically not interfere. We just observe and then measure these metrics. And then during that time, we'd be developing all of our models. And then for the 1 month period between May and June of 2022, we did kind of a silent implementation where we started to try out our optimization model, but we weren't recording any metrics. All we did was we ran the model, gave the recipe to the milk bank staff to implement, and just started to get some feedback from them, which we used to fine-tune things. And then on June 1st of 2022, we did the intervention period. So this is phase 3, where we ran the optimization. At that point, we didn't touch the optimization model anymore. We didn't touch the machine learning model anymore. We just simply asked it to create a recipe every day. We gave that recipe to the milk bank staff, and then they took it away from there. And then at the end, and so that lasted for about 5.5 months. And then we kind of had a cool down period at the very end where we just simply observed them going back to the baseline period. We weren't using optimization anymore. And we're just observing how they were doing things and just measuring any changes to the baseline metrics. Okay, so before I show you the results, I just want to give you a bit of context of what was happening in the milk bank at the time. So on the x-axis is time. You see on the far left, November 2021, which is when we started this trial implementation. So this graph shows the total volume of milk in inventory at the milk bank by week. Okay, and so what you see at the red line, which is when we started, the milk bank at that point was actually at a historic high in terms of milk in inventory. So about 1,600 liters. They said they were swimming in milk. I hope they were not literally swimming in milk, but they had a lot of milk. Okay, so they were at about 1,600 liters, and then at the yellow line in March, they made the decision to switch to pasteurizing 7 days a week. So up until that point in time, they were actually only doing it 6 days a week, and they said we have so much milk, we'd like to be able to do more. We're actually going to switch to 7 days a week. And so all of our modeling, all of our assumptions were based on a 6-day-a-week schedule. And then we said, whoa, wait a second. Like, don't forget, we have this, experiment running here, and everything was predicated on 6 days. And they say, yep, that's true, but we're running a real milk bank here, so we're going to do this, and you guys are going to have to roll with it. So I said, fine, we will roll with it. And so between the yellow line and the blue line is when we had to kind of retrain our models and adjust things and tweak things and do extra simulations to make sure it all worked. So we could have a 7-day-a-week model by the blue line, and then we trialed it out for a month to do some tweaks, and then from the green line onward is when we had our optimization period. And then as you can see, while the milk volumes were quite high in phase 1, basically as they went to 7 days a week, you can see the milk volume decline almost linearly after that, to the point where later in phase 3, in October of 2022, there were a few weeks where the milk volumes were so low that we couldn't even do 7 days a week of pasteurization anymore. We switched to 5 days a week. So just the takeaway from a contextual perspective for this slide is that in phase 3, when the optimization model was live, it was in a sense more constrained because it just had less milk to work with. Okay. The other piece of context that I want to give is this figure. So this figure shows the average volume of a donation coming in the door. Okay, so for every deposit that comes into the milk bank's door, on average, how many liters of milk is in that donation? So again, you see in phase 1, it was relatively high. It averaged about 13 liters. And then right before phase 2 started, there was this precipitous decline in the average volume per donation to the point when it went from about 14 down to 8 liters in the span of about one to 2 months. And then from that point onwards, it started to tick up again slightly, but it still hovered around 8, 9, 10 liters, which obviously much lower than 13. And so why is this important? There was a constraint I didn't quite tell you about, but in the pooling model, there's a constraint that says if you use a donation in a batch, you should use at least 5 liters, just so that we don't have lots of small volumes going into a batch, because that adds extra workload for the staff. And so again, just the contextual takeaway here is that it's much easier to get at least 5 liters of a donation into a batch when the average donation volume is 13 compared to when the average donation volume is 8. Okay, so yeah. So despite the fact that these two slides show, what seem to be some potential headwinds, our results were actually positive, and I'll show you that on the next slide. But before I do that, I just wanted to point out one other thing on this slide. So you see this precipitous drop around April of 2022. And so why is it that the average donation volume had such a steep decline at that point? If this was live, I'd ask the audience a question. But given this is a webinar format, I'll just tell you the answer. The answer is because the COVID restrictions lifted. And so if you recall, at this point, Ontario was, I think, in its 3rd wave. And we were under a lockdown during that period. And that's exactly when it lifted. And we were joking around. We're like, oh, haha, the moms were sitting at home with nothing to do but pumping lots of milk. And then as soon as the restrictions lifted, they all went out again. And then, the donation volume just kind of went down. And so we thought it was kind of a funny, joke. And then they actually asked the donors why. And then that's exactly what they said. They said basically because the restrictions were lifted and there were less restrictions around movement and going out again. And so they just spent less time pumping at home. Okay, so despite some of those headwinds that I described, the results in the phase 3 compared to phase 1 were actually quite positive. So comparing phase 1 and phase 3, we made about 140 recipes in both phases. This fat pass rate, so this would be the fraction of pools that met or exceeded a target fat level, which was 3.6 grams per deciliter. And so that moved from about 44%. So at that point, only about 4 in 10 batches passed that threshold. We went from about 44% to 57%. So that's about a 30% relative increase. For protein, similar. So the fraction of pools that met the protein bar was 53% before it went to 70%. So again, roughly another 30% relative increase. I think this is the most impressive number, the simultaneous pass rate. So the fraction of pools that met both the fat and protein pass rates, both the fat and protein thresholds. Previously, it was 1 in 4, and we got it close to 1 in 2. So about a 76% relative increase. In terms of variability from day to day, that's the fat MAD and protein MAD. MAD stands for mean absolute deviation. So just think about that as the variability in the fat and protein levels from recipe to recipe. So fat went down about 10%, protein went down about 30%. And then how long did it take to make the recipes? So previously it was about 7 minutes on average. We got it down to about 3 minutes. I mean, it's still, in both cases, it's not a lot of time, but the important thing is the computer is hopefully making quicker but better, as well as better pooling decisions. Did we waste more milk in phase 3? We did not. bear in mind that from the previous figures, we had less milk to waste in phase 3. So that one was kind of a wash. Did we lose more milk to bacteria or failures pre-post pasteurization, pre-pasteurization or BC post-pasteurization? So we didn't. Basically, those failure rates were identical. And then the last thing is, did we just get lucky in phase 3? Did the donors just give us more fat-rich and more protein-rich milk in phase 3 than what we had in phase 1? And so these are the average and standard deviation of the fat and protein by donation. And so statistically, they're indistinguishable. They're basically the same quality milk in both phases. Okay, so I think that might be it for time. So I'll just summarize really quickly. This was an optimization driven, sorry, a data-driven optimization framework for donor milk pooling that is specifically focused on use without an analyzer. Of course, this could still be used for milk banks that do have an analyzer, because the analyzer portion is where the machine learning comes in. If you have an analyzer, you can still use that, plus the optimization model that we have that comes up with recommendations for recipes. At the same time, some milk banks that we've spoken to say they would love to be able to get out of the analyzing game if they could, because that's a huge workload issue. And so this is another thing to consider. We are trying to do permanent implementation with our milk bank in Toronto. And we are also speaking with other milk banks elsewhere to try and build partnerships with them who want to try and implement something like what we've tried in Toronto. And if and when we are successful, at least in Ontario, hopefully about 1,000 preterm infants will be receiving this nutrient optimized milk recommended from our model, which, as was described by several presentations previously, strongly linked to improved outcomes. Okay, thank you.
Whoa. That was just fantastic. It's clear that science is now the limitation. What Tim has shown is we can begin precision nutrition for premature babies in the NICU by guiding the milk composition computationally. Those tools are here. We need to start building. I can't think of a better outcome for IMGC than to take 20 years of knowledge and start funneling it into the algorithms that Tim is developing. Fantastic. All right. So I know you've been sitting, standing, watching. We're going to take a bio break. 5 minutes, and come back in 5 minutes and we'll start the panel, the meeting of your dreams. You get to ask questions of all these world experts in your favorite subject. We'll see you in 5 minutes. All right. You've had your bio break. Now it's possible to answer any question, put in the Q&A chat, and we will ask the panel the questions. I'm going to sneak in a quick one. Nicole, knowing what is possible now as capabilities from analytics to computation, what would you like to have in your NICU as first priority to help babies right away?
Probably the ability to optimize the donor human milk and know what we're actually giving to babies. Because we use donor human milk and it's kind of like an afterthought that it might be insufficient. Like I was saying in the presentation, when we get the serum routine labs and the sodium is low, for instance, or the growth is faltering over time. And I don't want to just blame it on donor milk. Donor milk saves lives and, but it's nice to know. And sometimes we actually will, if there's not mom's own milk available in some cases, and we know that the baby is getting mostly donor milk because, like, let's say the mom went back to work and is not pumping, or, COVID restrictions are lifted and she's going out and not pumping anymore. No, just kidding. But sometimes we'll switch from donor milk to formula sooner. And maybe we don't have to do that if we can optimize the donor milk. And even testing the sodium in the donor milk, we can see, is it inadequate or not? So we just really don't know what's going on when we get low sodium. We just, a lot of times, just blame the donor milk when maybe that's not the case. Maybe it's the diuretics, or maybe it's something else going on with the babies. So I think optimizing donor milk would be the first thing I would want to have. So I'd want to work with Timothy and
Okay. Yeah, I think we're all for that. Actually, I'm going to ask Lindsay a question from the group. Uh, what you're doing is magnificent, and it's highlighting just how variable milk composition is. And two questions, basically, I think, rolled into one. One, is it possible, knowing what you know about these analytical capabilities, how big of a project would it be to be able to put the kinds of analysis that you describe into Tim and Nicole's hands? And ultimately, could you imagine this being something that even mothers themselves could do? And I'm going to ask you, and then I'm going to ask Daniela the same question. Oh, Lindsay, you're muted.
Thanks to Daniela, we now can measure, we know the methods for measuring these micronutrients, and they can be done relatively rapidly because most of the work was developing the methods and validating the methods. And Daniela is, I won't speak for her, but I believe she's very happy to collaborate in this regard. But it does need mass spec. I mean, I don't know that there are any key nutrients you could look at, but it does need a mass spec to do that. Not that the facilities that the colleagues on the call are working in don't have that, they do. But I don't see how mothers can do it, frankly. Yeah. Daniela?
Yeah, I mean, if you want to analyze anything basically at home, you would need to have something that does not need basic analytical knowledge, but you need an instrument like, collect milk, put it in, press a button, and we get the magic numbers. If we would have this, you wouldn't need me. So I don't think that is really possible at this stage. Maybe Tim can develop some algorithm generally for these things. I'm a basic researcher, so I wouldn't be the machine learning person to ask for. But I think if you want the detailed information that we can get now, you can't do that at home, certainly not with someone who's not trained to do so. To find some key things like sodium, if you can focus just on that, I can think of some just basic wet chemistry stuff you could basically, do, but you would still need some sort of lab tools, I think, and, know how to do it. But there are ways for some things. I think if you want a comprehensive panel, you will have to go to a lab to do so.
Oh, yeah. Now I have to ask Tim. So, Tim, Daniela brings a really good point forward that both Lindsay and she have been thinking about. If you had to measure a subset of analytes and develop capabilities to do that, are there patterns that you see in the analyses you've done so far that says, okay, these correlations are sufficient to predict, or do we have to put in place something that really does measure everything?
Yeah, it's a great question. So I agree with Daniel. I don't think we're replacing humans anytime soon, but presumably that day is coming, sadly, or scarily. I mean, not to be too much of an AI enthusiast, but I think there are things that these models can predict pretty well, with given the right data. Now, if you give it junk data, of course, it's not going to do anything. And so the biggest effort here is really around collecting a good data set that the model can learn from and then make inferences from, right? And I think there's 2 points there. So one is, how much data do you need? We talked about this a little bit earlier, Bruce, right? Like, do you need millions of data points? Like some of these AI models you hear about, in the public media or in the news around all these applications, they're trained on millions and billions of data points. How many data points were the models that I presented trained on? Hundreds. So we had a couple hundred data points, a couple of hundred donations and samples and measurements of fat and protein, and then we could generate this model. And so then the second point that I just want to make is you can predict anything. It's just a matter of how close to accurate the prediction is going to be, right? How accurate the prediction is going to be, how close to the truth. And so to give you a sense, our protein predictions had about a 10% mean absolute percentage error. So what that means is, let's say the average value in your data set was like 0.9 grams per deciliter. That means on average, we're predicting between like 0.81 and 0.99, let's say. And because of the long tail, that means the majority of the predictions are within that interval. Like more than half of the predictions are within that interval. And so it depends on what you're going to use it for, right? If you're using it for very precise fortification needs or very precise, something else, then that might not be enough accuracy for your goal. But if you're using it at a higher level and you're mixing it with a bunch of other things, that level of accuracy could be sufficient for your needs. It's really dependent on what you're going to use it for.
Okay. Another question that I think goes directly to Nicole, but I'd like everybody's opinion. It's fortifier. And of course, the perfect would be a human milk-based form fortifier. But that raises its own challenges. Could you imagine a liquid versus powdered human milk fortifier? And what would that represent as a challenge for NICU? Is it better to go powder or liquid in the NICU? And making powder, does that lose activity?
So I'll jump in and go first. And this kind of goes with Dave Dallas asked a question on the chat too about, does powdered human milk-based human milk fortifiers exist? So not at this time, but Sarah Taylor and colleagues are developing a reverse osmosis powdered. They basically take human milk. So you all may have seen this, and so concentrate it and could be in a powdered form. But as we know, it would need to be tested, and safety is a big issue for any new product. But I think safety in general is the issue for the powdered human milk fortifiers because of contamination, whereas liquid fortifiers can be sterilized. And so that is the main difference. So there is an inherent risk more with the powdered fortifiers, but volume displacement is incredible. I have a slide that I didn't show because I didn't have time, but the liquid fortifiers displace 20 up to 30% of the milk given at each feed. So the human milk-based fortifier right now, as most people probably know, is has the most volume and displaces the most. And if you fortify all the way up to 30 kilocalories per ounce with the liquid human-derived human milk fortifier, it can displace 50%. And the real challenge with these fortifiers is we don't really know what's truly in them, right? We know the basics. You can look at the nutrition label. But for instance, with the two bovine-based fortifiers, I've talked to Dave Dallas about this too. There's more of an amino acid based and more of a peptide based, but I don't know what peptides the babies are getting because I don't know what enzymes the industry uses. And I would like to know that information. And I would say a lot of my colleagues don't even know the differences between the whey-based bovine-derived human milk fortifiers and casein-based. We also don't know what the degree of hydrolyzation does. So we're just so far behind. And I think that with us in the NICU knowing everything about the medications we give these preterm infants and everything about TPN, we really are behind on enteral nutrition, especially with these human milk fortifiers. I want to know what I'm giving the babies. And for me, it's more of a justice issue right now. So yes, a lot of research needs to continue in this. And I think powdered fortifiers are probably the way to go to answer your question, Bruce, because they don't displace as much of the base milk volume of either the mother's own milk or donor milk. But right now it's just a guessing game in enteral nutrition for preterm babies.
Yeah, it's clear we need more Tims. Every neo should have a Tim. Lindsay, can I ask you to wade in on that? You see astonishing differences among mothers. But one of the themes is that early milk seems to be, in general, a little bit more nutrient rich. And that implies that if you are getting milk for NICUs, you'd want to be encouraging mothers to share milk early, which is difficult. But is there something about the milk of a healthy mother that says, "Oh yeah, um, this for whatever reason is super milk"? Oh, muted.
I think that it is probably largely a process that is developing from leakiness among the mammary cells and more nutrients leaking into the milk than something specifically designed for the baby. A few nutrients actually go down, especially minerals, in that first month. So it's not completely consistent. But I do believe that nutrients, as well as other constituents, a lot of them, what is there when the baby is really small, his stomach is tiny, that is absolutely critical to make sure that what's there in the first few weeks in human milk is mirrored in the preterm milk. I mean, the stomach is just amazingly small at that period of time. Right. They say an apricot, the size of an apricot. And so it's very critical to take that early period into consideration. And we at least will publish and be coming out in Advances in Nutrition as a series of 7 papers in a few weeks, so that the percentiles for all the nutrients in that first month will be in that publication as well.
Whoa. How about sodium? Nicole brought up sodium, and I had not realized that that sodium was an issue for NICU babies. Um, did you see much variation in sodium levels, Daniela?
Oh my. I would have to look into the data. I know it was, I mean, it's a macronutrient, so it's pretty abundant in our milk, which was term and healthy. So that's not necessarily an indication of what preterm milk will look like, I would assume. But honestly, I am not sure right now if we saw a big decline or a big increase of sorts. I don't recall anything outstandingly like crazy variation, but I would have to put up the slide again. Honestly, at this point, I'm more for like, ooh, I like sodium, it's easy to analyze in my world. So, but you know, it was like 4,000 samples. I don't remember, honestly. Lindsay, do you?
No, I don't. Could look it up quickly.
Yes, I will.
I
I will
believe
check,
it was,
Bruce.
I believe it was flat throughout the whole of
I thought so.
lactation. Yeah. Right,
Not
you
a
let them
problem.
know, Daniela.
Yes, I am on my way.
But we're also interested in mastitis, and that is an indicator of mastitis, which we're looking into as well.
Actually, yeah.
Right.
Tim, did the various NICU or the various pooling systems were they capable of seeing things like mothers with unusual compositions driven by things like mastitis? Could you pick that?
Yes. Yes, we can.
Okay.
We can look at that. Yeah, and a surprising number of women did have mastitis in some countries.
If
We also have cytokines and other indicators of infection, along with sodium potassium ratios. So somebody will be doing a lot on that.
I did see the slide now on sodium. It's a little bit higher in the beginning and then it really easily flattens out. I mean, if we want to show it, I can share that. But I mean, generally,
Yeah.
it was,
Because I think one
yeah.
of the things that, I mean, Tim's presentation was so provocative that if we could know more and guide more mothers as donors and direct that towards specific infants, then I think Nicole's work shows we could do better for each infant. Then the question becomes, what do we need to measure? And some of the things that we imagine measuring, yeah, will require mass spectrometry, et cetera, but maybe others fast and furious. Tim, was there anything that jumped out at you to say, oh my gosh, these mothers are unique for this reason? Even if, as an engineer, you weren't able to say, okay, the lactocytes are under stress, but is there something that just computationally jumped at you?
Um, My short answer is the short answer is no, I don't think so. But I think it's because we weren't really looking for that. We weren't looking at that. But I think what I would say in response, and it builds on something that Nicole mentioned at the end of her presentation, was around personalization. And so I think, if you can decide, what is important to measure and what actually has an impact on outcomes, right? And if we're not matching the right, nutrition to the right baby right now, then once you know what it is that you want to match with them, right, we could potentially develop, kind of similar models as we did for fat and protein, but for other things, to make sure that, maybe not everyone is getting the same because the needs are heterogeneous, right? Our focus was trying to essentially create as uniform a product as possible, high quality and uniform, because we know fortification was going to happen on top of that. And if we produce a product that isn't uniform before fortification, it's going to not be uniform post fortification either, right? And so uniformity assumes something about the population, but maybe to your point, maybe personalization is the way we really should go in the future. But that's certainly getting out of my realm of expertise.
Well, and I think just to include Carlito's vision in the discussion, virtually every rich male in the world knows their cholesterol level. Cholesterol is a painstaking molecule. It's a pain in the behind to measure. It's not very predictive of anything, and yet the world knows it. And so if we could prioritize key things that matter for babies, then we could imagine a cholesterol education program analogy for babies. And so now I want to ask the panel, if you were going to take a TIM optimization strategy, combining all things, what would we want to be able to measure and then inform every mother about for her baby and for her donor milk, so that not a single milk delivered to a baby in a NICU by Nicole wasn't just right for that baby? What would be the cholesterol of babies? Lindsay, I got to ask you first. The sheer irony is I'm watching my screen, and 30 seconds ago, what popped up on my screen is from my bibliography server, new publication from Lindsay Allen. So
Okay.
I don't know whether my machine is watching this or it's just a reflection of how important and impactful Lindsay is. But so Lindsay, what's the cholesterol of babies?
Um, Not sure, possibly protein. One thing we're going to do next is analyze the predictors of milk composition. Is it the mother's genomics? Is it her diet? If it's diet, then we can probably get a good picture of mother's dietary quality by looking at a few key nutrients, such as riboflavin, for example, which is very responsive. Vitamin B12, which is always a worry of mine since some women have low intakes and so on. That doesn't exactly answer your question, but I think we need to better understand why there is a huge variability that you're noticing. It's unbelievable. Is it nutrient transporters, genomics? We're working on that. So Camilla Arrington is working on that whole part of the study, Carmen Arrington, and we'll know a lot more in 2 years. So big variability, what's causing it, we honestly don't know. I really don't think it's all diet, but if you look at those low values in Bangladesh, you can see that usual maternal status is an incredibly important thing. So anyway, I would say at least one marker, B vitamin, maybe riboflavin, vitamin B12, which is not easy to measure, unfortunately, and then a macronutrient such as protein or maybe energy. And of course, we can measure lactose and protein and fat with the infrared analyzer very rapidly and quickly, which is what I assume people are doing. So anyway, but we don't know which nutrients predict growth yet. That's the problem. All the supplementation trials are, there's maybe some small benefit to infant growth, but not much at this point that we can put our fingers on. So we need more of those or better analysis of what's been done, which is what we're trying to do.
Yeah.
Yeah.
So again, imagine that we could draw on the resources of the world, build the analytics, deploy the analytics, and provide it to individuals as knowledge. Daniela, you've been you've been running milk till you're blue in the face. What would be the one thing you'd say, this is what we should do? Even if we're going to do more later, what would be that cholesterol education program that everybody says, yeah, I'm healthier now because of this? What would make every baby healthier?
Uh, as Lindsay said, I think there is nothing we can just put our fingers right now on that say this particular part of milk is the most important one to get that cholesterol where we want it to be, to get all the micronutrients where we want them to be, get all the macronutrients and energy where we want it to be. I also don't think we can look at this separately. So I do think Tim is not incorrect if we say, unfortunately, the humans will be replaced at some point, and we'll have a very good prediction with all the data we accumulate these days and also what has been accumulated in the past. I mean, this is very challenging, so you would need a lot of data for the machine to learn very good predictive models. But I do think also if you want to do what we maybe can do now, it would be some rapid analysis maybe for some key biomarkers. Like we have the mirrors, or we use personally the Unity version for one reason and another. I just don't see right now how we can quickly do a panel of micronutrients like in 5 minutes. It's just not going to happen if you want to have the detailed data. So we would have to go back and figure out what can be done rapidly and is useful in this whole context, given like, oh, just the B vitamins, they're all energy contributor in that sense that they are all involved in the metabolic process of energy. So you want to not look at those cofactors who may be a basis of, getting that energy out there for the baby into the milk. So I honestly, unfortunately, I don't have that magic bullet. If so, I would have so much funding I could retire on. I think for us as basic researcher like me, who are really on the lab focused, collaborating with someone like Lindsay, who knows more of the nutrition than I ever gonna be happy. If I know half of it, I'm gonna be very, very, very knowledgeable. And then going forward to someone like Tim, who can use all of this and throw it, I'm sorry, Tim, into the computer and Python and R, whatever you guys want to use, to make some sort of use of it to see what can there be done that can be done quickly. That doesn't need $1 million setup in the lab. Well, let's guess more like $5 million if you want to have everything in there, plus the person who knows to run it. I mean, you guys like to get the data. I appreciate that. I love to get data. Data is great, but the time and effort it takes to get to the data that I am at least comfortable of what I give to you is a whole nother part of research no one really appreciates other than the person who has actually been in the lab to see it. But I mean, data is great, but you want to be confident in the data. So that's a whole nother issue of analyzing milk in the first place. I know it's a long way around it. I'm sorry, but I can talk to it like for hours and we can all sit together and might help or not, but I won't have the bullet for it.
Let me just add, don't forget milk volume.
Right.
In any study, the volume of milk that an infant consumes is one of the main determinants or predictors, at least, of growth. There's always the chicken and egg issue, and that bigger infants consume more milk. So, to unravel that is a real challenge. When you do the preterm feeding studies, I guess the baby can consume ad libitum, but that isn't necessarily the case out in the real world. And certainly we can measure mother's milk output. It's a very sticky area because some women presume, and Davis only produced 400 milliliters a day, and yet their babies seem to be fine, and others produce 1,200. So there's another big mystery, but we mustn't forget it. It's not just composition, it's how much of it the baby gets.
Yeah, and even that we don't have a good handle on. Weighing the babies before and after, weight, I mean, it's all very crude, and it's 2025.
Well, we used deuterated water, so we did it the gold standard way, but that's measurements 7 times over 15 days each time you do the volume. But we have good data, and that's in the papers too. Yeah.
Oh yeah, and I think you illustrate the conundrum that this workshop is about. We want to accelerate analytics and developing ways to do it, and then turbocharge those analytics into point of care. And so for Nicole, same question: what would be the cholesterol of babies? And also, why is sodium such a problem for babies mechanistically? What's going on?
So I would vote for protein at first because, like I was saying in our ping study, we presumed that the protein was at least 1 and it was 0.6. And some protein from certain milk banks can be greater than 1. So, like Tim said, we have to know the base that we're working with because when issues with growth arise at the bedside and we're rounding on these patients, the first thing we point to is the base milk, either mom's own milk or donor milk. And both are presumed to be 20 kilocalories per ounce, by the way. And we know that's not true. But protein is the biggest issue that most neonatologists will supplement. And each NICU deals with this donor milk low protein situation separately. My previous institution, they just used to add liquid protein to all the donor milk. But then if you think about what you're doing, you're taking a product that is human derived and should help with tolerance and should have a nice balance of whey, protein, and casein for these babies that are high risk, like not just preemie babies, but cardiac babies, babies that have malformed guts, like gastroschisis. And we're assuming we can use the donor milk. But if something is being added that has a bovine product from the beginning, and different types of protein as well, we're changing the composition just because we assume that it's not good enough. So I would go back to Tim and say, I would love to know that the donor milk is consistent, reliable, that I don't have to blame it for everything that's happening with the baby's growth or intolerance, and then go from there. But I would really like to know the protein first. And then, sorry, what was your second question on that?
Yeah, do we know why sodium is mechanistically linked to such adverse outcomes? It's just sodium.
Yes. So the problem in preemies is they don't absorb it. So their kidneys aren't really developed and able to do what they would normally do in a term infant or you and I until 34 weeks corrected gestational age. So their kidneys are to blame for most of the issues. And also, a lot of the medications that we give to babies have sodium in it. So at the bedside, we don't actually talk about what the true sodium load is. We can measure serum sodium and urine sodium, but there's not good standard values of what a urine sodium should be for a baby. And serum sodium it really fluctuates. For instance, one of the most common antibiotics we give to our preterm babies is called ampicillin, and that has a lot of sodium load in it. But when we're talking about babies at the bedside in terms of their sodium levels and their nutrition, a lot of us don't consider the extra sodium load that the babies are getting in their flushes when they have to flush the lines. They use sodium. Like I said, in medication delivery, we're using sodium. So it becomes a big issue. And then when the babies have issues with their lungs, we give them diuretics, which that makes the kidneys act very strange. So that is why it's important to know what the base milk we're giving has in terms of the sodium content. And that's something that I can't do right now. And there was a question on the chat too about, do we use sodium point of care at the bedside? I don't personally know of any NICUs that use it at the bedside to inform nutritional practices. But as Lindsay talked about in mastitis, it's used, and my previous colleague used it in Florida to look at the onset of secretory activation. But that was more for a research study where when the sodium was the correct level, they would inform the moms that were pumping that they were entering secretory activation. So I think we do need to think about this at the bedside because the donor milk is getting blamed for a lot of low sodium and a lot of low protein, when in fact, maybe it has an okay amount in it.
Oh. Well, and so I'm going to.
Actually, yeah, I think
Okay.
we found levels in milk were lower than were assumed by the National Academy's output, for example, so that they may be an overestimate of requirements.
Yeah, this sounds like it's not only a preemie issue, but a specific preemie. I mean, this is where the precision medicine rubber meets the road. Is if a baby's getting diuretics, then yeah, I would think that's got to go into Tim's algorithm. And the ability to say, here's the baby's chart, here are the milks, boom, solution. And I'd like it coming into a tube in 20 minutes. So yeah, Tim, I have to ask you, since you are a little bit back of the babies, but have the broad sense of engineering and a pragmatic view, and I'm going to say, if one had my fondest dream, you are now head of Health Canada, and you could say this is a priority. Canada needs to know what to measure in milk to make babies healthier. Yeah, what would you measure?
Oh, I worry that I don't have the right nutritional expertise or background to answer this question. Based on what I understand from my far more knowledgeable colleagues, I think I'd probably double down on something like protein, as Nicole mentioned, because we've used that, and that's one of the reasons we use protein as kind of the proof of concept of our approach due to its importance around neurodevelopmental outcomes. But I'll say, to answer your question a different way, if I was in charge of Health Canada, I would think about how to lower the barriers to innovation in getting new ideas to be implemented in practice. Like I understand why it's important to have all the safeguards and do all the rigorous testing. But I feel like we have a bit of a culture of, probably well justified, a culture of, conservatism around safety, which is obviously important. But I think often that lends itself to slowing down the implementation and of kind of new and novel discoveries. So I would try and find a balance there.
I love that. There's a question from the chat, and I'd like to go after Lindsay first. But so you didn't interrogate every diverse population in the world, but in just 5, you saw pretty impressive ranges. And in fact, one could even imagine them clustering separately, iodide in Bangladesh, etc. And we tend to think of adverse outcomes. Hypothetically, as it has been seen with the development of autoimmune diseases, are there populations in the world that the infants seem, for whatever reason, to be unusually successful?
Unusually successful
In
considering
any criteria, protected
maternal
from
diet.
allergy, metabolic disease, obesity, type 2 diabetes, physiological, anything that you say, that seems unusual. I wonder if that's related to the fact that their milk composition is so what. Knowing how much of the potential for lifelong success in immunity, in metabolism, in physiology, cognition, all of that is in this early window of development. And milk is so critical. Do we see system level immunity, physiology, metabolism variations that coincide in any way with what are pretty striking compositional differences in the milk when you look as carefully as you have?
Since we haven't analyzed around the world yet, nobody has. That's an impossible question that will be coming up for answers. I would sort of argue that, when we did these samples, we screened for a reasonable quality diet of the mothers and no supplementation to start with. And I would say that looking at the 50th percentile, they were astonishingly dissimilar across the countries in Africa, Copenhagen, Bangladesh, and so on, except for the couple of nutrients, B12 and also, iodine, but that's because of universal salt iodization, that difference. B12 and vitamin D.
Right.
And that has to do with the nutritional status of the mother very clearly. Danish babies grow hugely. You know that. And why that is, we don't know, but possibly is nutrition. They did have slightly more calcium in their milk, which was interesting because that's something we always thought was not different. And I believe they had slightly higher protein as well. But it was such a challenge that when you looked at weight for length, those Danish babies were so long that they had low weight for length scores. And so we had to eliminate some from the study for low weight for length because they were so huge. So there were those sorts of issues going on. So I don't know of any. I use Denmark as an example of obviously very healthy, long babies. And let's look more closely at the milk composition. The volume wasn't very different, not per kilogram body weight, which of course you need to correct for. So I don't know. I'm really excited to learn about the genetics of the milk volume and the genetics of the nutrient composition. And that will be very key to understanding everything better. Yeah.
Yeah. All right, so we only have 10 minutes left. And I wanted to give each of you a chance to ask the others questions because the genius of milk is its diversity. The complexity of milk is its diversity. So, opening up, you can ask any of the other panelists anything, including asking them to put an area on their agenda.
All right, Nicole, when are we going to start to collaborate?
Let's do it soon. I think a barrier actually to collaborating on milk banks is just the willingness of the milk banks to reveal their processes and practices. And I've found that that was the case in some of the publications on donor milk. So you don't want to demonize, any product. And so I think we'll have to carefully discuss how we want to proceed. But yes, we should definitely collaborate. I think Canadian research is amazing, and you have access to so many babies and so much information. And I feel like in the US, we kind of function in silos, especially at each NICU.
Yeah, I'll say there's only 4 or 5 milk banks in all of Canada, so it's much, in a sense, a little bit more centralized. And especially if you compare to like Europe in the far end of the spectrum, right? Many more, but smaller ones. But I think you're right. Like our experience in talking with several other milk banks, a couple in the US, a few in Europe. I think the biggest factor is just the kind of whoever the lead is, whoever the medical director is, or whoever's running it, that they are kind of open and willing to try something new, right? Because they see like that maybe there's some part of the current process that they think can be improved, or they just want to try something better for the babies, right? That I think has been the biggest determinant of who we've connected well with. So, yeah.
Yeah, Tim, I wanted to ask you, so the trend in April 2022 was that, do you know if that was the same for other milk banks around the world and in the United States when did it correspond to when they lifted their
It's a
COVID?
good question. You're talking about the average volume decreasing from
Yeah.
donations? Yeah,
Hmm.
I don't know. I would be surprised if other milk banks didn't see something similar. Of course, when lockdowns happened and did happen or did not happen, get lifted, not lifted, that will change from geography to geography. But I'll say a lot of people, when I ask people why they think that happened, people might recall that summer there was also a formula shortage. So people
Hmm.
are kind of naturally go towards, oh, maybe it had something to do with the formula shortage, but I don't know. I assume you saw this as well, but incidentally, for people, donations went up when the formula shortage happened because people were more motivated and it became more of a top of mind issue for people. So had the opposite.
Yeah. Yeah. Actually, interestingly, the Florida Milk Bank, they're the only milk bank in Florida, but their activity and calls really increased with the formula shortage. So people were trying to get milk from that milk bank, and that was probably true of a lot of the milk banks. Um, but yeah, just clinically, we're thinking of partnering with our OBs to try to increase milk donations. I think that, it's great and, what a value that these moms can pump extra milk and help preterm and sick babies by donating. But I mean, it's a resource that I think we need to tap into a little bit more, right? Like it's just a lot of women, lactating women who have extra milk don't realize that they can donate and that there's certain things that would disqualify them but a lot of them probably do qualify and would. So when moms just deliver a healthy term infant and get discharged from the hospital, some of the OBs will put a little blurb about consider milk donation. Maybe we need to talk about it more on social media, even put some signs on the highway, consider milk donation. So that's something to think about as well moving forward. So you wouldn't have that limitation and your algorithm of the volumes being limited. I think we can really increase it. So this is a huge controversial topic. Like, do we have a limited supply of donor milk in the world, or can we tap into more resources? So yeah, to be continued.
Daniela.
Okay, just to chime in, not because I'm such an expert in donor milk, but we collaborated with Marianne Perrin, and I think Nicole at least knows papers of hers. She had that grant for the DHM study, and the study protocol has been published a few years back. So we did a bunch of analyses, basically everything we did for milk, we did for the donor milk she shared with us, and there's HMO data and all. I don't know if there's any kind of volume data in terms of what was donated to make pools as well, but there's their outcome papers. I know 3 were submitted, so there should be more information coming out at least on this front as well. That might be useful to inform and create smart studies later on for human milk, and maybe helps shedding a bit of light into at least some of the nutrient compositions from these milk banks. That was done worldwide. So there are a few from the US and then at least one from Poland, I remember, and some others where you can hopefully then also show the differences or maybe even some processes. I'm sure Marianne's happy to answer questions if she can too. If you want to get in touch with her, she's very nice if you haven't met her yet.
Yeah, that would be wonderful. I just wonder how many of the milk banks participated, because in order for me to do my study with the Florida Milk Bank, I had to submit an application. I had to promise that I wasn't trying to develop any new products. And I just don't think any of the banks want to be targeted. That's why in my presentation, I didn't even say where we got the milk from. And so what I've learned is, even though Himbana has about, I think, 33 milk banks, their processes are very different. But yeah, if that study included all of the Himbana milk banks, I think that would be the most helpful. And just breaking those barriers, saying we just want to know where we're at so we can improve, and we're not trying to make anybody look bad. I think for me, like entering this research space, as a clinician now becoming a physician scientist, I'm just really surprised about the barriers there are in working together. Because I just thought if the milk banks were a part of a big collaborative, everybody would work together really well, but it just doesn't always happen like that. And I think money is a big barrier too. I know to run these milk banks, it takes a lot of personnel, it takes money. And like Tim said, even like for his project, they didn't change the workflow, but to change the workflow of a normal day, Monday through Friday, eliminating holidays and the milk bank, just adding one more thing can really slow the process down, maybe cause safety issues, decrease efficiency. So I think that needs to be considered. I think most of the milk banks use Holder pasteurization in the US, but even proposing a different type of processing, they would need new equipment, they would need to train new staff. So I think I'm more cognizant of these issues when I tried to say, like, let's change things. It's easy to say that, but I think we need to start really at the foundation of working together and getting all the information from all the milk banks. This probably works better in Canada, I'm guessing, Tim, but I'm not sure.
Well, if it was up to me, every high school would have a course on babies. And you would teach young people all of these issues. And at a career decision moment, this would be part of their decision making. I think it would change everything.
They do. It's called sex ed, but it's not the type of education that you're talking about.
Yeah, they probably need assistance with that too, but yeah, I'm delighted to welcome back Jennifer Smilowitz. Jennifer.
Yep.
Thank you. Thank you. Okay. Hi, everyone. I'm back. Wow. What a riveting set of talks and discussions on the challenges, but we also provide solutions, right, for diagnosing milk for improved clinical applications. Okay, so please note, there is a recording of today's webinar and it's available on our website around July 1st. So stay tuned for an email from us. Just click on the 2025 Ross Workshop tab and you'll be able to watch the online seminar or webinar. So the topic for this year's Ross workshop is proposed by attendees from the previous workshop on the exit survey. That's why we're asking you to complete this year's. But the speakers for the workshop are selected by the IMGC Scientific Advisory Committee, or SAC. And I would like to give a big round of applause to thank this committee for researching the topic and selecting such an incredible group of speakers. Okay, now as a nonprofit organization, the IMGC heavily relies on funding support by sponsoring partners who help pay for our 4 platforms. So I'd like to thank our 2025 elite sponsors whose generosity has been instrumental in supporting our advancements in milk science. Here they are. Okay, and finally, scientific advancements could not be made without the brilliant and passionate innovators and scientists that makes up our base, and that's all of you. We rely on your partnership in advancing the field, and our success is dependent upon your insight, your input, and engagement. There are 5 ways you can be involved in our organization. So first, if you aren't already, subscribe to Splash Milk Science Update. Again, our bi-monthly publication on milk science for human health, and please share the articles with your network. That includes your parents, your partners, just your friends, everyone you work with, your students. Join our LinkedIn page to network with fellow milk science experts and enthusiasts from all other fields. Learn about job opportunities, funding, and internships, and events in the field. And next, please, please, please complete the 5-minute exit survey for today's workshop. This is so important because your vote will help us identify the topic for next year in 2026. And you can also engage with IMGC in various working groups. So we have co-author partnerships and review papers on specific milk science topics that you can work on with us as a consortium. Now, if you're interested, let us know on the exit survey that you'd like to participate in some of these activities. And finally, I hope to see you in person or virtually during our 22nd annual IMGC Hybrid Symposium, co-hosted with Arla in Aarhus, Denmark. That's on September 16th through the 18th of this year. You can find information for the symposium on our website. And thank you all for participating today, and we will see you soon. Bye.